So Giuseppe Lippi is a full professor of clinical biochemistry and molecular biology at the University of Verona in Italy and director of the laboratory service of the University Hospital of Verona in Italy. He has published over 1,800 articles in peer-reviewed journal. His total impact factor is over 6,800 and the Hirsch index is 100, something similar to myself. In 2017, he has been appointed as Secretary of the European Federation of Clinical Chemistry and Laboratory Medicine. He currently chairs the task force on COVID-19 of the International Federation of Clinical Chemistry and Laboratory Medicine. The stage is yours, Professor Lippi. Thank you.
Thank you. Okay. Dear friends and colleagues, it's my real pleasure and privilege to be invited to this virtual edition of the Pre-Post Analytics in Laboratory Medicine Study Day, with the presentation on the leading pre-analytical issues in COVID-19 diagnostics. I really wish I could be there with you, but I guess this won't be possible for such a long time. Before specifically addressing the main topics of this presentation, let me briefly introduce some aspects of this novel pandemic disease. Coronavirus diseases are not really new for humanity, since COVID-19 is the third coronavirus outbreak that has occurred over the past 20 years, following several acute respiratory syndrome in 2002-2003 and Middle East respiratory syndrome in 2012. The most striking difference is that the two former outbreaks have caused altogether less than 2,000 deaths, while COVID-19 has already affected nearly 39 million people worldwide, causing already 1.1 million casualties. This number is far below the over 50 million deaths occurred during the Spanish flu pandemic in 1998 and 1999. But it can be easily calculated that the ongoing COVID-19 outbreak could be responsible for nearly the same number of deaths if appropriate preventive and contention measures will not be undertaken all around the world. As concerns the responsible pathogen, the International Committee on Taxonomy of Viruses has concluded that the microorganism causing COVID-19 is a beta coronavirus, belonging to the same family of viruses that may cause flu-like syndromes and common colds, and that has been finally called Severe Acute Respiratory Syndrome Coronavirus 2, which is abbreviated to SARS-CoV-2. SARS-CoV-2, as all other coronaviruses, is an enveloped virus with positive sense single-strand RNA genome, encoding 4 leading structural proteins, which are known as spike, and which also contains the receptor binding domain through which the virus binds to its natural receptor at host cell surface, the envelope protein, the membrane protein, and the nucleocapsid protein. The genome of SARS-CoV-2 also contains additional genes encoding accessory proteins, such as the RNA-dependent RNA polymerase, which is essential for viral replication. Before specifically analyzing the main pre-analytical vulnerabilities in COVID-19 diagnostics, I think it may be necessary to provide a quick update on COVID-19 diagnostics. According to the World Health Organization, a confirmed case of COVID-19 is a person with laboratory diagnosis of COVID-19 infections, independently from the presence of clinical signs and symptoms, which thus places laboratory diagnostics at the very core of this pathology. The routine diagnostic confirmation shall be based on detecting unique sequences of viral RNA using nucleic acid amplification tests, such as RT-PCR, while nucleic acid sequencing can be used for confirmation only when this will be necessary. Optimal diagnostics shall then consist of an amplification test based on the last 2 independent SARS-CoV-2 gene targets selected among the IN, E, IS, and RNA-dependent RNA polymerase genes. The IFCC Task Force on COVID-19, which I have the pleasure to chair, has also endorsed interim recommendations on molecular testing. Briefly, insufficient evidence still exists for suggesting the definitive advantage of selecting molecular assays targeting a specific SARS-CoV-2 gene. However, it seems reasonable to recommend that these assays should be based on not less than 2 gene targets, preferably employing 1 primer capable to detect the gene shared by most coronaviruses, including SARS-CoV-2, combined with a second set of primers which more precisely detect one SARS-CoV-2 specific gene target. Due to time constraints, I will not be able to deeply describe the many methodological assays that are now, that are now available for molecular diagnostic of SARS-CoV-2 infection, whose details can, however, be found in this interesting article, which also highlights the advantages and limitations of the different analytical techniques. As concerns the reference specimens that shall be collected, these are respiratory materials and include either upper respiratory tract specimens represented by nasopharyngeal and oropharyngeal swabs, or lower respiratory tract specimens represented by sputum and or endotracheal aspirate or bronchoalveolar lavage, especially in patients with severe respiratory disease. Although upper respiratory tract specimens still retain the preferred samples for detecting SARS-CoV-2 RNA, the diagnostic accuracy, especially the diagnostic sensitivity of RT-PCR in these specimens, appears far from optimal. Cumulatively, the sensitivity in nose specimens is between 50–60%, while that in oropharyngeal specimen is even lower, between 30 and 40%. Combining naso and oropharyngeal specimens, the final sensitivity approximates 70 to 80%. The low diagnostic sensitivity of RT-PCR in upper respiratory tract specimens seems attributable to a myriad of pre-analytical and analytical problems related to sample collection and analysis, which will be briefly summarized in one of the next slides, but is also due to the biological characteristics of the virus. As you will see in this figure, during the first day of infection, when the patient is incidentally still symptomatic, the viral load in upper respiratory tract specimen may still be lower than the limit of detection of the method. The same situation occurs at the end of the infection for a period that can last for 1 to 2 weeks, when viral shedding at lower intensity is still present, but cannot be detected with many molecular biology assays. In both these two windows, there is a high risk of generating false negative results when the diagnosis will only be based on molecular testing. It is thus not surprising, as shown in this interesting study, that the sensitivity of RT-PCR for detecting SARS-CoV-2 in naso and oropharyngeal specimens is directly dependent on the number of sequential samples collected and tested, increasing from 86% with one single test up to 93% with 2 consecutive tests, and up to 100% only after the 6th consecutive test. This obviously implies that patients persistently testing negative despite high likelihood of bearing SARS-CoV-2 infection, which can be either due to contact tracing or because they have suggestive symptomatology, shall then be subjected to short interval repeated testing before they could be definitively labeled as negative. One aspect that we need to make clear from the beginning is that the quality of testing for COVID-19 diagnostics is unavoidable, as much as for any other human diseases, or even more. This concept is based on the clear-cut evidence that diagnostic tests may generate inaccurate results in 2 ways. First, a false positive test result would erroneously flag healthy subject as being infected, with many avoidable consequences, such as undue quarantine, unless contact tracing or unnecessary diagnostic of treatment with drugs, oxygen, and so forth. The second risk is that of producing false negative results, which is no better than the former for clinical and the societal consequences, since uninfected persons may have treatment delayed, contact tracing cannot be started, and especially when asymptomatic. The person may not be quarantined and would then contribute to spread the virus among relatives or even within the community. A paradigmatic example is shown up here, happened just 2 days ago with pro cyclist Michael Matthews, who had to pull out from the Tour of Italy for a likely false positive case. It has also been clearly established that many pre-analytical and analytical factors may have a deep impact on the diagnostic sensitivity of molecular testing of SARS-CoV-2 in upper respiratory tract materials, including, for example, identification errors, inadequate procedures for sample collection, handling, transportation and storage, sampling of inadequate material for quality or volume, presence of interfering substances, manual errors, specimen contamination, or even tests carried out in patients taking antiretroviral therapy. As concern potential analytical errors, this may include tests performed outside the diagnostic window, active viral recombination, which may then impair probes annealing, the use of non-sufficiently validated assays, lack of harmonization of primers and probes, as well as instrument malfunctionings, collection of insufficient or inadequate material for testing, non-specific PCR annealing, misinterpretation of expression profiles, and use of non-clinically validated diagnostic cutoffs. I will show you now some paradigmatic examples on the importance of the pre-analytical phase in COVID-19 testing. One of such cases is presented in this article, where the authors demonstrated that inadequate nasopharyngeal sampling carried out by untrained operators in the presence of nasal obstruction was the case of false negative RT-PCR test results in 4 patients with SARS-CoV-2 infections, since all results became then positive when swabs collected few hours afterward by a skilled otolaryngologist. Doubts also remain about the efficiency of self-sampling, as shown in this article, where patients' home-based self-collected foam swabs only display 73 accuracy, 75% sensitivity, and 72% specificity compared to reference flow swabs. The discrepancy was especially evident for specimens with low viral load. A slightly better agreement has been found in this study, where self-collection of anterior nasal swabs for SARS-CoV-2 molecular testings display 86% and nearly 100% negative and positive agreement compared to nasopharyngeal swabs collected by healthcare personnel. A latter agreement was observed by comparing self-collected saliva sample with healthcare staff collected nasopharyngeal swabs. In this study, which encompassed accurate patient training to self-collect the nasopharyngeal swab by watching a 2-minute video, as in the figure in the right of this slide, the agreement between self and healthcare staff collected specimen was as high as 96%, and the overall diagnostic sensitivity was not significantly different between either technique. It is hence undeniable how this evidence will emphasize the extreme importance of providing accurate pre-analytical training for increasing accuracy and safety of this type of testing. An intriguing approach for harmonizing swab collection is the automation of this activity. Some prototypes of throat or nasal swabbing robots have already become available. These automatic swab machines are equipped with vision systems which record the facial structure and thus enable to recognize the right points to swab in throat or nasopharyngeal pathways. According to manufacturers' claims, the arm of the robot collects the material for laboratory testing by safely placing the swab in nasopharynge or throat for an appropriate time, and then places it in a jar and screws the lid. This would enable to reduce the risk of cross-infection and save precious time and resources, since the hood swabbing procedure could be completed in 5 minutes compared to the 10-15 minutes, which would be necessary for a conventional procedure carried out by healthcare personnel. However, the lack of clinical validation studies remain a major drawback for routinely using this practice. Another interesting example of the impact of pre-analytical quality on SARS-CoV-2 molecular diagnostics is reported in this interesting investigation, where the SARS-CoV-2 positivity rate using molecular techniques in upper respiratory tract specimens has been assessed at 2 time points, early in the morning before washing and later in the afternoon. Interestingly, not only the cycle thresholds appear to be lower in the morning, but also the rate of positivity was found to be considerably higher than in the afternoon collected samples. This may be due to the fact that common activities during the day may influence the local accumulation of virus in the upper airways, so that the detection rate would fall accordingly as the day progresses. Continuing to describe the critical value of pre-analytical quality in SARS-CoV-2 molecular testing, this study shows the impact of chlorhexidine mouthwash on SARS-CoV-2 viral quantification in saliva. As clearly demonstrated by the figure, mouthwash with chlorhexidine was associated with a significant reduction of SARS-CoV-2 viral load in saliva for up to 4 hours after gargle. Mixing specimen before or after extraction is another variable that shall be considered for the quality of sample pooling diagnostic strategy. That is especially important due to the current challenges in purchasing reagents. In this study, it has been demonstrated, for example, that pooling specimens after extraction would be associated with an essential increase of the cycle threshold compared to the native sample, and thereby with potentially reduced diagnostic sensitivity, thus underpinning that pooling specimens before RNA extraction would be more advisable. Another paradigmatic example of potential pre-analytical error in molecular testing of SARS-CoV-2 is described in this interesting report. Briefly, this short note emphasizes a case of oligonucleotide probes contamination during production caused by synthesis of full-length control oligonucleotides, which span from forward to reverse primers in concomitance with another probe order. This report thus highlights the importance of establishing thoughtful quality control assurance, even within a context of reagent shortage and delay associated with huge volume of testing during a pandemic. As concerns the possibility the new emerging variants of SARS-CoV-2 may have an impact on the diagnostic performance of nucleic acid amplification tests currently used in clinical laboratories, the authors of this article have calculated the mutation rate of SARS-CoV-2 genes, underpinning that the S and E gene seem to be those mostly non-conservative. So it cannot be discounted that the current molecular assays for detecting SARS-CoV-2 genes may be losing accuracy as genetic mutation within the original viral genome gradually accumulate over time. In this interesting study, the authors have identified the alignments of binding sites of 33 oligonucleotides using different RT-PCR assays for detecting SARS-CoV-2. And they have found that up to 79% primer binding sites were mutated in at least one viral genome. It seems so reasonable to hypothesize that some primers using these assays may now be inapt to detect some viral variants, thus reinforcing the recommendation of continuously optimizing oligonucleotide sequence. A paradigmatic example is reported in this study, which found that a specific recurrent mutation in SARS-CoV-2 may impart the capability of detecting the virus with E-gene amplification in a commercial RT-PCR method. However, despite the E-gene amplification fail, the commercial assay still correctly identified positive sample for SARS-CoV-2 due to the use of a dual target approach, encompassing also the amplification of ORF1ab. Interesting data are also emerging on the potential use of saliva instead of the reference nasal and oropharyngeal swab specimen. For example, in this study based on 156 samples, 50 of which were positive for SARS-CoV-2, the concordance with virus detection using the same RT-PCR assay in saliva was excellent, displaying 96% sensitivity and up to 99% specificity. Notably, one residual sample tested positive in saliva, but not in nasal and oropharyngeal swabs. In this other article, for example, the authors not only found that the overall RNA viral detection rate was 10% higher in saliva than in nasopharyngeal swabs, but the SARS-CoV-2 titers in patients with COVID-19 may also be even higher in saliva than in nasopharyngeal swabs, so that the sample matrix may even yield better detection sensitivity throughout the course of infection. An interesting evidence on clinical validity of using saliva as specimen of choice in SARS-CoV-2 diagnostics has emerged from this study, showing that devices combining the prevention of drops generation and expansion together with saliva stabilization may be seen as viable means for non-invasive self-collection of clinical specimen by untrained subjects, thus allowing RNA stabilization for up to 1 month at room temperature. The compelling need of enhancing the efficacy, encompassing both throughput and rapidity, of molecular testing in certain areas and under some critical situations is however challenged by some many practical issues. Last but not least, the potential shortage of reagent and personnel, as I said before. This is clearly reflected, for example, by the findings of this important survey carried out by the American Association of Clinical Chemistry, and showing that as for the end of September 2020, over 50% of worldwide clinical laboratories are facing a dramatic shortage of test kits and reagents, with over 70% of them reporting paramount challenges to further increase their testing capabilities. The concept that the ideal testing volume cannot be reached in many worldwide countries, including highly developed nations, has been reiterated in this editorial published in the British Medical Journal just a few days ago, and concluding that the first priority of each national government shall be to guarantee that everyone who needs is tested, and that test result can be delivered within a reasonable 20 hours turnaround time. However, there is clear evidence that this strategy is not currently being met. Taken together, the current evidence would so testify that the ongoing COVID-19 strategy testing is failing, since the various bottlenecks in test accessibility are causing enormous delay in generating results, so fostering a vast array of clinical, societal, and economical consequences. Some interesting strategies have so been proposed for enhancing the efficacy of molecular testing, encompassing the estimation of positive test probability, using kits without RNA extraction and purification, sample pooling, and/or rapid antigen testing. I specifically concern rapid antigen testing, the impact of the different functional sensitivity of a conventional molecular test, and the rapid antigen assays is summarized in this figure, showing that both the diagnostic windows and the so-called gray zone could be different using one approach rather than the other. Briefly, the diagnostics window of conventional molecular tests is broader, allows to identify patients infected by lower viral loads, and thereby the chance of obtaining fast negative results extends over a shorter period of time before acute infection. On the other hand, it remains to be clearly established that the real infectivity of patients with SARS-CoV-2 viral load detectable by conventional molecular tests and not by rapid antigen tests. Are these really infected? Is some molecular testing really more globally effective than antigenic analysis? Only the future will tell us. To conclude my presentation, I would finally like to draw your attention on the results of this interesting study, which further confirms the enormous diagnostic potential of humans' best friends. Briefly, the authors trained 8 dogs to identify saliva or tracheobronchial secretions from patients infected by the virus. Notably, the diagnostic accuracy of these scent dogs was 95%, which is much better than molecular testing using upper respiratory tract specimens, with sensitivity and specificity as high as 83% and 96%. Therefore, we can now speculate that a dog would only need between 10 to 100 viral particles to detect SARS-CoV-2, even in the asymptomatic phase, compared to the over 80 million viral particles needed for a conventional molecular assay. Again, only the future will tell us if this new type of testing will be successful or not. What we can be sure, however, is that this possibility will open new highways for investigating canine pre-analytical training. Thank you for your attention.